Welcome everyone to Iris Biotech's 12th online workshop. True to our slogan, Empowering Peptide Innovation, we're keen on knowledge exchange and getting in contact with scientists all over the world. Therefore, we started this workshop series in summer 2021. Today, our focus is on fluorogenic amino acids for solid phase peptide synthesis. Our two guest speakers, Dr. Fabio De Moliner and Professor Mark Vendrey from the University of Edinburgh, will present a fascinating way how to introduce and apply small fluorogenic amino acids in peptide synthesis. My name is Raimund Meyer. I'm working as a COO at Iris Biotech, and today I will guide you through this workshop. As always, I would like to begin with some technical information. To ensure good data transfer of the presentation, all participants are automatically muted and your video cameras are switched off. This live event is recorded, thus if you enter late or miss anything, please check on the bottom left side of your Teams screen if you are watching the recording or if you are live, which is shown by the red broadcasting symbol. You have the possibility to pause the video, rewind, or jump back to the live session. In addition, the recording of the workshop will be uploaded on our homepage afterwards. Questions can be submitted by using the Q&A chat of Microsoft Teams. You can enter your name or submit your questions anonymously. Initially, your questions are just visible for the organizers and will then be published to the audience. So don't worry if your submitted question does not appear on the chat screen immediately. If you have any technical queries, you can also submit them in this Q&A chat, and our technical assistant will then connect with you and try to solve the problem. I would now like to quickly give you an overview on today's agenda. We will start with a short company and topic introduction by our Chief Marketing and Sales Officer, Karin Rustler. Afterwards, our external speakers, Fabio De Moliner and Mark Vendrey, will present to you the introduction and application of small fluorogenic amino acids in peptides. As mentioned, questions will be collected throughout all talks in the Q&A chat and are forwarded to the speakers. But of course, you can also get in contact via email or phone call after this workshop. Let's now continue with the first agenda point, a short introduction to our company by our CMSO, Karin Rustler. Karin obtained her doctoral degree in organic chemistry at the University of Regensburg, and beginning of 2020, she joined Iris Biotech as a project manager. And in the meantime, she's now our Chief Marketing and Sales Officer. Karin, thank you for your time. Please go ahead with your presentation.
Thank you, Raimund, for the kind introduction, and also welcome from my side to today's workshop. It's a pleasure to see that so many of you are still interested in our topics and that this type of online events, which was basically started due to the coronavirus pandemic, is still of interest. As Raimund mentioned, I will start with a brief company introduction for those of you who did not know Iris before. And for those of you who already know Iris, don't worry, I will keep it short. For this slide, I just want to focus on a few aspects. The company Iris Biotech has been founded in 2001. Our portfolio comprises more than 6,000 different products for which we are working together with different manufacturing partners around the globe to realize demands from milligram to kilogram quantities for research purposes up to GMP quality. At our site in Magdredwitz, we have an analytical laboratory, and for synthesis, we have a separate laboratory in Willstedt, also in Germany. This also allows us to provide a custom synthesis service. For those of you who never heard of Magdredwitz, I can highly recommend visiting the beautiful so-called Fichtelgebirge, of which we are surrounded by. On this slide, you see a picture of our company building, where we are operating from and where your orders are being processed. As mentioned briefly in the beginning, on the one hand, our mission is to support our customers in their ongoing projects from research scale to commercial production. In this regard, we are mainly active in the field of peptide synthesis and modification by providing amino acids, resins, reagents, and solvents. But we are also active in related fields such as linker technologies and products for drug delivery. But for us, our slogan, Empowering Peptide Innovation, does not only mean to provide innovative products, but also to act as scientific advisor by providing and exchanging knowledge. In this regard, we built up our knowledge base, which includes topic-specific brochures and flyers, which can be downloaded from our website. Besides, we are publishing our bi-weekly newsletter, in which we highlighted previously the fluorogenic amino acids suitable for SPPS. And with this, I would already like to thank you for your kind attention and pass on to Fabio and Mark, today's guest speakers who invented this technology. Thank you.
Thank you, Karin, for giving this introduction. Yeah, let's now go on with our highlight and switch to the external guest speakers, Dr. Fabio De Moliner and Professor Mark Vendrey. Fabio obtained his PhD in organic chemistry from the University of Genova in 2010 under the guidance of Professor Andrea Basso. Then he joined the research group of Professor Christopher Halm at the University of Arizona in Tucson to focus on the design and application of chemical methods based on multi-component and domino processes. In 2015, he joined the Vendrey Group in Edinburgh, and his main research interests there are the development of optical probes for in vivo imaging with a focus on the preparation of fluorescent amino acids and the labeling of small metabolites. Mark Vendrey graduated in chemistry at the University of Barcelona in 2007, then joined the Singapore bioimaging consortium to work with Yongtae Chang in synthetic fluorophores for non-invasive optical imaging. And in 2012, he started his independent career as MRC academic fellow at the University of Edinburgh. Now he is professor and personal chairholder of translational chemistry and biomedical imaging at the College of Medicine and Veterinary Medicine in Edinburgh. His team has pioneered the design of activable fluorophores for high-resolution optical imaging in live cells and tissues, published in over 120 papers, and built an ambitious research program linking physical and biomedical sciences. Professor Vendrey has won numerous awards, the latest being the Alfred Baader Prize by the Royal Society of Chemistry in 2023. Fabio and Mark, it's a great honor to host you as our guest speakers. The stage is yours. We're looking forward to hearing about your research. And for the audience, please post your questions by using our Q&A chat.
All right. Good afternoon, Raymond, Karin, and thank you very much for organizing this workshop. It is really a pleasure to explain you a bit more how we've developed this technology that is already commercialized by Iris Biotech, and hopefully people will understand a bit more what fluorogenic amino acids can do for peptide chemists. I don't know if the slide is passing. Yeah, here we go. So this is an outline of our presentation. We will do this as a double act between Fabio and myself. I will give you a short intro on fluorescent chemical probes, just to introduce what our research group does. Then Fabio will go into the details of the fluorogenic amino acids and how they are synthesized and their optical properties. And then I will finalize by explaining you how we use these fluorogenic amino acids to to synthesize fluorogenic peptides on solid phase peptide synthesis, and really briefly show you two case studies. And I will wrap it up with kind of giving you a very brief overview of the advantages and the limitations of this technology over other existing technologies. Striving to move on to the next slide. Okay, so fluorescent chemical probes. Our research group is specialized in the synthesis and application of fluorescent chemical probes. Some of you in the audience may be using fluorescent probes for your research. As chemists, you may know that there are many different types of structures that can be used in probe development. They can range from traditional like heterocycle-based fluorophores to peptides, which is going to be covering more on this talk, and also small molecules or larger constructs from nanoparticles to dendrimers or antibodies. So there's really a diversity of chemistry that can be used for probe development. And today we're going to mostly talk about peptides and amino acids. Then the other bit that we wanted to kind of explain straight away was this concept of fluorogenic amino acids. What is a fluorogenic amino acid? How is different from a fluorescent amino acid? To explain you in the simplest way possible this concept, we actually borrowed a picture from a Scottish painter. His name is Richie Collins. And Richie actually drew this picture of the Edinburgh skyline. For those who have never been to Edinburgh, this would be the castle of Edinburgh in the middle of the town. And traditionally, when we've been using fluorophores, and if I'm going to ask you to particularly look at, let's say, the sky of this picture, we've been trying to use fluorophores that would have the largest amount of photons being emitted, right? So just to be able to capture every single detail in that sky. You can see that if you're trying to spot as many stars as possible using those always-on fluorophores, or fluorophores that emit photons constantly, whereas you can see very bright stars, you may also struggle with areas where there is too many photons, right? This is kind of a limitation that we can address by using not always-on fluorophores, but what we call fluorogenic probes, which are fluorophores that would only emit a photon when they are in contact with a particular event. In this case, we try to match that fluorogenic emission with the binding to a specific protein or a target. When you do that, you can see that because all the probes wouldn't really be bright all the time, and they would only emit kind of when they are binding to a target, you can really spot stars that would be really hard to spot on a very bright sky. So hopefully this gives you a bit of an idea on how we are trying to maximize contrast when we're using fluorescence imaging by using these fluorogenic probes that are not always fluorescent, but they only fluoresce when they are really binding to the target. To have another visual of how this process happens, we took this movie where you can see two cuvettes here, and these two cuvettes have actually fluorogenic dyes in them. You can see that the left cuvette is showing a fluorescence orange colored emission, whereas the right cuvette doesn't really show any fluorescence. And I'm going to play the movie now, and what you're going to see is how this fluorogenic event happens, how this right cuvette, when it is in contact with a target protein, the molecule that will activate the fluorescence, it will become brightly green, and it does happen in really nanoseconds. And you will also see how the orange cuvette will turn into yellow, showing that not only we can switch them on and off, but we can change their colors. So hopefully the movie will play now. So you're going to see a standard fluorogenic turn-on effect, which is what we see in an off-to-on fluorophore, and then, well, the movie is cut, but you would see how this orange cuvette would turn yellow too. So this is giving you an idea that these processes, the off-to-on in a fluorophore, are really, really quick, and therefore they can be matched with a biological event so that we can see biological processes in real time. Okay, this fluorescence activation, as we would call it, requires fluorophores that are unique, and we call these fluorophores environmentally sensitive fluorophores. So these need to be fluorescent molecules that respond to their environment, so that they are not always fluorescent, but they are only fluorescent in specific conditions. We've put here some examples of commercial fluorophores that are environmentally sensitive, and just I've highlighted two rather broad categories, like pH-sensitive fluorophores and like the pH Frodo or Oregon Green. These are structures that would only fluoresce at specific pHs. I've also highlighted some other fluorophores that are called lipophilic fluorophores, or fluorophores that would be sensing the polarity of the environment. And two very good examples are Nile blue or Dansil dyes, right? And these are heavily used dyes in fluorescence imaging. So then when we wanted to start integrating this concept into peptides, then we thought, we wondered ourselves whether we could make environmentally sensitive fluorophores, but that could be easily incorporated into peptides. If you remember the structures in the previous slide, those were conventional dyes. They didn't really have amino or carboxylic acid groups. They would be difficult to put within a peptide sequence. So we were looking for fluorophores that would have a large change in their fluorescence emission, so that we would really maximize that contrast, and that would be built as conventional building blocks for a solid phase peptide synthesis. And ideally, we also wanted them to be small, so that when we incorporated these amino acids into peptide sequences, we would be able to retain the bioactivity of the peptides that we are labeling. And I think that this is, I'm going to move into the next slide, hopefully. Sometimes it gets frozen. See, here we go. Yeah. So I will pass the button to Fabio, who will explain you about fluorogenic amino acids.
Thank you, Mark. So thank you all for attending. And what our group has been working a lot with in this vein is to prepare derivatives of different amino acids with different fluorophores that were environmentally sensitive. And we've tried to cover different amino acid scaffolds as well as different wavelengths. As you can see in these slides, where we try to organize and summarize most of the fluorogenic amino acid that we developed in the latest year, we focused first of all on the tryptophan amino acid, which we have heavily and diversely functionalized with body P fluorophores by means of carbon-carbon couplings, either directly on this phenyl ring, which belongs to the fluorophore, to the body P fluorophore, or by means of triple bonds, conjugated double bonds. We've also functionalized with the tryptophan with fluorophore other than the body P. We've worked with MBDs, with these extended structures that are hybrid MBD fluorophores, and with silicon rhodamines as well. We've also investigated functionalization with fluorophores, in particular body P fluorophores in this case of phenylalanine to make fluorogenic phenylalanine analogs. And then we've moved on, and we'll be talking more in details about these specifically later on the functionalization of DAPA, the aminopropionic acid, with different NBD kind of fluorophores with different heteroatoms and different connecting atoms. Last but not least, we've recently developed and worked with analogs of tyrosine, where the fluorogenic amino acid has been realized by connecting body P cores with the tyrosine backbone by means of the diazo group with diazonium chemistry, which is quite well known and established old chemistry, but still very useful. Fluorogenic amino acid, actually, as I was talking before, stemmed from functionalization of the body P fluorophores that was connected to tryptophan amino acid by means of CH activation coupling. This is a palladium catalyzed coupling that was performed under microwave irradiation conditions in the presence of acid in acidic environment. And that allowed us to connect the body P fluorophore to the tryptophan functionalization on the indole heterocyclic core of tryptophan to generate the tryptophan body P. That was actually the first one of the first generation fluorescent amino acid that we developed. So going back to tryptophan body P, our first fluorescent fluorogenic amino acid, we found out that in aqueous environment, it's very, very little, it displays very, very little fluorescence thanks to a mechanism of photoinduced electron transfer in between the body P fluorophore and the indole core of the tryptophan amino acid. When tryptophan body P though is placed in hydrophobic environment, it lights up and its quantum yield rises to up to 20%. So on the same vein though, we then work to modify tryptophan body P to tackle some of the issues that are most important when it comes to peptide synthesis, solid phase peptide synthesis, and the design and generation of biologically relevant active peptides. So first of all, the body P core tends to display low resistance in acidic media. And our first improvement was to move from tryptophan body P to what we call tryptophan body P plus. Basically, this analog relied on the replacement of fluorine atoms on the body P core with cyano groups. And these cyano groups, thanks to the formation of a complex with the TFA or acetic acid or other acidic species, can stabilize further the body P core, which is way more resistant in acidic media, and so it's way more friendly to solid phase peptide synthesis. We then
Fabio, sorry if I'm interrupting you. We can still see your slide number 9.
Oh.
In full screen. So I think you already switched in between the slides.
Yeah.
This is not good. Apologies for the technical issues.
Maybe if you leave it like, well, now we see it in full screen again. Yeah.
Yep.
We are now on page 10. Yeah. And now it seems that the swapping also works.
Yeah. Yeah. Can you see slide 12?
Yeah.
Perfect. Thank you.
Thank you.
So the next issues that we wanted to tackle was the scalability of the synthesis and the size of the fluorophore. Functionalization of the tryptophan amino acid with a tryptophan core, even if the body P is not a particularly large fluorophore and fluorescent tag, it's still rendering a quite different fluorescent amino acid compared to these native analogs. So we then moving on to using the potem amino acid that rely on smaller NBD-like fluorogenic tags. And besides the size, Also, the scalability of the synthesis of this fluorogenic amino acid was greatly improved. In fact, the synthesis of the tryptophan body P core relying on a body P fluorophore requires sometimes quite messy chemistry, and the final coupling relying on microwave condition is also more difficult to scale up. Regards the synthesis, it's easy to notice how the selenium MBD fluoride, as we call it, which is the smallest labels in the POT-M synthesis, is 246 Dalton. And if we compare this to an Alexa fluor, and this is not one of the biggest Alexa fluor, the molecular weight is 653. Even visually, the difference in the size of the tags and so to the potential disruption to the native function of the peptide is quite striking. So, addressing the synthesis of the POT-M fluorogenic amino acid builds upon our previous work on the Scott fluors. The Scott fluors are fluorophores that are based on the MBD core, where by means of the replacement of the bridging heteroatom core from nitrogen to sulfur to selenium and to carbon, with quite simple chemistry, 2-3 steps from commercially available starting material, we managed to produce a platform of small conjugatable, orthogonal, and tunable fluorophores, which displays different fluorescent properties, all based on the nature of the bridging atom. As you can see here, the POT-M fluorogenic cores display different wavelength and are environmentally sensitive. They display bathochromic behavior. They shift emission moving from more lipophilic and apolar solvents like toluene towards aqueous media. The synthesis, in particular, of the selenium and the sulfur POT-M cores, which are the ones that we employed to prepare fluorogenic amino acid that are compatible with peptide synthesis, is actually quite simple. In the case of the selenium, we can start from commercially available and cheap chlorophenylenediamine. Selenium is inserted by means of simple reaction with selenium dioxide under reflux. Nitration follows, very classic chemistry condition with strong acid. This is all very scalable up to gram scale. And the final, the core endowed with a fluorine or a chlorine halogen is very suitable for displacement by means of nucleophilic substitution with amines that can happen easily at room temperature within minutes. When it comes to the sulfur analog, the synthesis is actually even smoother and shorter. We can start, again, even on gram scale, from commercially available dibromo-thiabenzodiazole. Reflux in nitric acid can lead to the nitrobromo precursor, which is slightly less reactive than the fluorine we use for the selenium synthesis, but still, under reflux in acetonitrile, can easily lead to amine derivatives, which are also fluorogenic. Coming to the synthesis of the proper fluorogenic amino acid, which are actually sold by Iris Biotech, we can see here how, starting from the fluorine selenium MBD precursor, a simple one-step reaction with FMOC protected diaminopropionic acid under mild heating in the presence of a base can lead, with great scalability, up to 500 milligrams in one single batch so far, can lead to the FMOC DAP and BSD, as it is sold by Iris. It's also possible, for the sake of practicality, to use mono-BOC protected DAPA. This is even more scalable, quickly reacts with the selenium MBD fluoride, and BOC can be quantitatively removed with the serine dioxane, and the FMOC group, which is key for the use of this fluorogenic amino acid in peptide synthesis, can be installed by means of a reaction at room temperature with the FMOC azide electrophile generated in situ. Exactly the same synthesis is applicable for the preparation of the sulfur analog, also sold by Iris. under the name of FMOC, DAP, and DTD. The bromide precursor is a bit less reactive than the fluoride, but again, upon heating in water methanol, the reaction is high yielding and scalable. And again, the same, upon heating, the POC amino acid can be installed and the POC protecting group can be swapped with FMOC upon deprotection and then installation of the FMOC with FMOC generated in situ. The two POTEM amino acids sold by Iris actually both display quantum yield around 20%. The sulfur MBD amino acid is emitting in the green channel. It has emission around 550 nanometer. The selenium one is slightly red-shifted with an emission wavelength emission maxima around 600 nanometers. And quite pleasingly, these amino acids are completely compatible with the most common conditions that are utilized in solid phase peptide synthesis. This is an example of HPLC study of the stability of the selenium containing analog. Purity higher than 95% was found upon treatment with 95% TFA in DCM, 20% piperidine in DMF, which are key in peptide synthesis, and also in the presence of TBAF, tetracis palladium, and hydrazine, that are among the most common reagents used for the deprotection of protecting group that you can find on side chains of peptides. And with that, I will hand it over back to Mark.
Yeah, thanks, Fabio. That's good. So, so now that Fabio has introduced how we make these amino acids, and we're going to concentrate on these two amino acids, the sulfur and the selenium ones, and we use these circles with the color so that you know that one is green emitting, the other one is orange emitting. After having made these amino acids, we wanted to see whether we could use them for solid phase peptide synthesis and whether they could indeed label peptides in a non-invasive way. So I'm going to present you briefly two case studies. So this is the case study one. The case study one is a project that was led by Fabio and Susanna here on the screen. And basically what we did in this project was we decided to label a relatively long or short peptide, like 20 mer peptide that is called 101A. Which is an alpha helical peptide that can form a very tight binding event with its pair sequence, which is the 101B. These 101A and 101B had been reported before, and so we just use them from the literature as kind of a model system to see whether we could detect this binding between 101A and 101B by just using one of our amino acids as the reporter, right? So the hypothesis was that if the 101A was labeled and labeled particularly with one of our fluorogenic amino acids, we would only see this fluorescence when 101A and 101B would be binding together, because that kind of binding event would induce an increase in the fluorescence emission. And to confirm that that binding was actually due to, or that fluorescence was due to the binding, we also labeled the 101B with a biotin so that we could have a second color to colocalize the two signals. So the three questions that we were trying to answer really were, well, first of all, can we synthesize the peptides by solid phase peptide synthesis using these POTM amino acids? Second, could we retain the structure and recognition properties after labeling? Will these peptides behave the same way? And third, can we detect an increased fluorescence emission when these two peptides bind with one another? So full disclosure of the chemical synthesis or the solid phase peptide synthesis. I just put here a note on the top left highlighting that this compound 12 that you see here is the NBSD amino acid, so that nobody gets confused. we just use a standard approach using tentagel resin, standard FMOL-based solid phase peptide synthesis. This is a sequence of 101A, as I mentioned earlier. Just to make things simpler at the beginning, we decided to put the amino acid as the last position at the N-terminal N. And you will see later that we have also made peptides where we have put the fluorescent amino acids in the middle of the sequence. And we did 3 kind of sequences: the non-labeled one, which we refer as acetyl 101A, the labeled with our amino acid, which is kind of this 5-101A. We also use fluorescein as a commercially available dye that is conventionally used for labeling peptides as a control. And I'll show you one piece of one experiment that we did comparing both. Just for full disclosure, we obviously made the 101B, including a biotin, and in the same way, this doesn't really involve the use of our pot M amino acids, but just that you know that we obviously made the 101B pair for our biological experiments. These are traces of how the crudes look after the cleavage. So I'm gonna just highlight here, for instance, this is the one using the pot M amino acid, the one that had this selenium atom. It's a relatively nice crude. Obviously, it gets better when you purify it, but it's not a bad crude to purify. Again, these are crudes that we get for the 101B, labeled with biotin, and the fluorescein, right? So we can see that the purity that you get from the crudes is definitely comparable to the one that you would get with other dyes. So the first answer, or the answer to the first question, whether we could synthesize peptides using solid phase peptide synthesis using our amino acids, the answer is clearly yes. The answer to the second question, or to answer the second question, which was how will this labeled 101A behave compared to the non-labeled peptide? Are we really altering the properties of the peptide? Because if we do, then that label is actually not as good as we want it to be. So we did this experiment, or Susanna actually did this experiment, which is a circular dichroism experiment where we compared the CD of the labeled peptide, which are the green lines, versus the unlabeled peptide, which are the blue lines, right? You can see that this is clearly an alpha helix. You can see the obvious peaks at 190 plus, the 208, the 220 plus. So this is clearly an alpha helical structure. And when you put the fluorophore at the N-terminal N, the alpha helical structure doesn't really change at all, right? So we retain that secondary structure after we've labeled. And we believe that this is because these amino acids are relatively small. As Fabio alluded earlier, they are no bigger than 1 tryptophan. And so if you put them definitely in the right place, there shouldn't be a major change in the secondary structure of your peptide sequence. Then the last question that we wanted to answer was, can we actually retain the recognition of 101A and 101B? We knew that these 2 peptides, the 101A and 101B, should be pairing one with the other when they are unlabeled, but would they still bind one another when we label the 101A? And we did this cool experiment where we actually use TERF microscopy to look at these binding events. So in the green channel here, what you're seeing is the fluorescence emission of 101A. You can see that there are some events where 101A is actually binding to 101B, and those are these bright spots that are just magnified in this square. And you can see that when this binding is happening, we can also see the signal of the Alexa fluor stress-davidin, which is the area that is binding to biotin. So we know that this induced emission of 101A is because the 101B is there. So the 101A and 101B pair is retained after labeling the peptide, which is what we needed. And the fluorescence increase is maximized when this binding event happens, as you could see from the background fluorescence that is much lower when those binding events are not happening. So if you look at this area where there is no binding, the fluorescence is pretty low without any washing, obviously, because there is no washing in this field of view. You can see an increased fluorescence when the peptide is binding to its target. I mentioned earlier that we did a fluorescent labeled peptide. And the reason why we did that was because we wanted to compare this ability to switch on and off that our amino acids have. This is a property that a fluorescent dye wouldn't have. And the best way to illustrate this is to show you how the fluorescence of these dyes changes with the environment. So in this graph, you have here the 101A in red, labeled with our peptide. And in green, you have the 101A labeled with fluorescein. You can see a trend of increased fluorescence emission when the polarity is actually changing. This is a common trend that we see in these environmentally sensitive fluorophores. You can see that fluorescein, on the other hand, wouldn't really sense that trend. So a fluorescent would give you a very similar signal throughout any environment that you would put the peptide to, and therefore this enhanced emission that we see when there is binding wouldn't be that strong, right? So that's what is giving us the contrast. To show you kind of another example of how this contrast is magnified and what are the working concentrations that can give you this contrast, we did this other experiment where we put the same amino acid and we actually modified it with a long acyl chain so that we could incorporate it into liposomes. And what you're seeing here are really the images of liposomes that have actually been labeled with our lipid modified amino acid. You can get very strong signal within the liposome, but even at those concentrations, you don't get any background signal when the dye is not part of the liposome. So that is basically telling you that even when you use 1 micromolar of dye, the dye would be background free completely unless that peptide is binding to its target. And this is something that is very difficult to achieve with really bright dyes like fluorescein, body piece, or Alexa fluors, which at 1 micromolar concentration would be extremely bright under any microscope. So the case 2 study that I just want to present very, very briefly is, well, can we actually use this way of labeling peptides to answer biological questions? And I borrowed this cartoon, which is a very complex cartoon. I'm not going to go through it from this paper by Keith et al. This is something that we did in collaboration with Matthew Horrocks here in Edinburgh, who is an expert in super resolution microscopy, in particular with a type of super resolution microscopy that is called peptide pain. This is a very good paper from his lab where he actually demonstrated for the first time that you can do peptide pain in live cell imaging. So the target that we wanted to image was this protein that I'm highlighting here, PSD95. I know that this cartoon has a lot of information, so I'm just asking you to look at these 2 keywords here. PSD95 is a protein that is key within synaptic transaction, and it's part of a larger structure that we call synaptosomes. So the synaptosomes would be the complexes that are formed between synaptic events, and PSD95 is a protein that is involved in the formation of these synaptosomes. So one of the questions that structural biologists want to answer about the behavior of this PSD95 is whether this PSD95 is kind of in a monomer form, or it can dimerize. And these are the questions that super resolution microscopy would be able to answer. But we needed to be to develop a labeling method for PSD95. The way that we decided to prepare a labeling method for PSD95 was to use this peptide ligand. So this is a heptapeptide that had been reported before that binds very tightly to PSD95. And then we felt that this was a good proof of concept for our technology, because labeling this heptapeptide with a conventional fluorophore would likely induce a major change in the chemical structure. So we thought, can we actually make a small fluorescent analog of this peptide by using our small fluorogenic amino acids? So as I alluded earlier, we can make solid phase peptide synthesis using our fluorogenic amino acids, but putting the amino acid as one more building block within the peptide sequence, not only on the N-terminal N, as I showed earlier. This is the sequence that we made where we just replaced the isoleucine amino acid that is present in the native ligand with our POT-M amino acid. We chose isoleucine because it was a more hydrophobic amino acid and the one that we thought could tolerate better this change in chemical structure. Again, full disclosure of the chemical synthesis, the HPLC trace of the crude before purification, relatively clean, purified, even cleaner. And these are the images that we got when we put this peptide in the synaptosome. So you can imagine that we isolated these kind of suborganelles from the brain of mice, and then we labeled those PSD95 with our labeled peptide. And what we could see were the clusters that were formed by PSD95. So this is an image that these bright spots correspond to PSD95 in these synaptosomes, and we could see that there are actually clusters of PSD95 that are formed with super resolution microscopy. I didn't have a scale bar here, but just to give you an idea, the smallest clusters that we can see are around 20 nanometers, which is within the super resolution microscopy range. So this peptide allowed us to see that indeed PSD95 aggregates in the form of dimers or in the form of larger oligomers. And this is something that couldn't be done before because we didn't have such a small peptide that would have a small label that would retain binding to PSD95. So it's a good example to showcase the power of this technology. Just to wrap up, I think that the applications of this technology are broad. They can be used as fluorophores on their own, and we've shown you some examples of that. They can be used as fluorescent amino acids, fluorescent peptides, but also we are working towards expanding these technologies to larger entities like fluorescent proteins, which would give us the possibility to target many different biological proteins or events. And we also prepared this last slide, which may be the kind of the most informative slide when it comes to comparing our technology versus existing ways of labeling peptides. I've put on the left the main features, good and bad, of the POT-M amino acids, and on the right, the main features of other off-the-shelf fluorophores. I didn't focus on a particular one, but just I wrote some general trends. One of the biggest advantages of the POT-M amino acids is that they are really small. And as Fabio alluded earlier, they are in the range of 200 to 300 Daltons, which is the size of a large amino acid. They are neutral, which means that you are not putting any extra charges into your peptide sequence, which can be important. This is something that outperforms the conventional off-the-shelf fluorophores because they can be much larger, and sometimes they include positive or negative charges, which can also affect the peptide sequence. They are relatively easy use. You can just treat them as any other amino acid when you're doing your peptide synthesis. This is in contrast to what you need to do when you are labeling peptides using commercial fluorophores, other commercial fluorophores, which are sold as succinimyl esters or malamides, which means that then you are often bound to including them on one single position of the peptide sequence. And this links to the third point. You can put these POT-M amino acids anywhere you want within the peptide sequence, whereas kind of an off-the-shelf fluorophore would be limited to either the N-terminal N or kind of to the side chain of a reactive amino acid. They have fluorogenic behavior. They can actually be used to detect direct peptide binding, and we've shown some examples of that. Off-the-shelf fluorophores, some of them can be fluorogenic. Most of them are always on, so you will need to add washing steps into your imaging protocol to be able to remove any excess of unbound peptide. Their main limitation would be their brightness. They have quantum yields and extinction coefficients that are not as bright as the ones that you may get with, for instance, Alexa fluors, which allow you to see very, very low concentrations of the fluorophore, like low nanomolar. Whereas the POT-M amino acids in those images that I've shown you, they were used at 100 nanomolar, 500 nanomolar. We feel that nevertheless, they are good for the kind of the peptide capabilities, because of those are the KD constants that you would get with most, with many peptides. So therefore, that the working concentration of the peptide and the brightness of the fluorophore are matching in the sense that they are both in the high nanomolar range. But this is something that you need to consider. If you want to label, for instance, a peptide or an antibody that would have really low nanomolar concentration, you may find yourself with not having enough brightness to detect in the microscope. With this, we'll just obviously acknowledge the work of the entire team that is behind our research, which are just, I listed them here on the left. The collaborators that helped us with this particular work in Edinburgh and outside, and the kind of the funding bodies that are kindly supporting our research. So we will stop here, and we will be happy to answer any questions that the audience may have. And we apologize again for kind of the technical issues during the talk. I'm glad that we could make it through the end. I'll leave this slide as kind of the last slide.
Yeah, thank you, Fabio and Mark, for sharing your results with us. During your talk, we collected quite some questions, and let me start with one that came in right before you started explaining that in this slide you are still sharing. This question: how bright are these fluorophores compared to the classical large-scale dyes such as Alexa and SLI dyes? So are they in principle suitable for cytometry and fluorescence microscopy, or is that brightness not enough? Or is it just a question of, which concentration you have to work with in your particular experiment?
Yeah, I would say they are bright enough for fluorescence microscopy. We have done a lot more experiments than the ones that we have obviously disclosed today. We've used them in fluorescence microscopy. We've used them in flow cytometry, and they are detectable in really the hundreds of nanomolar range, I would say. If you look at the paper published, you can see that the concentrations used in the 100 to 500 nanomolar. This is kind of a standard concentration for peptide imaging. I think that I think they are definitely bright enough to be seen. We are just kind of being very honest upfront and saying that if you wanted to detect a peptide that was at perhaps 1, 5, or 10 nanomolar, then you may need to rely on additional contrast to be able to see the signal. But I would say 95 to 99% of the experiments that any peptide chemist would do with fluorescence would be absolutely fine.
Mm-hmm. Then there is a rather physical question. Can you explain why the Stokes shift between excitation and emission wavelength is rather big? It was in a range around 100 nanometers compared, say, to other classical fluorophores.
I'll take this one then. It is a property of the nitrobenzodiazoles. So fluorophores, they have unique properties when it comes to their electronic transitions, and those are defined by the chemical structure of the fluorophore. The Stokes shift of these nitrobenzodiazoles is generally quite large, as the person highlighted. This is something that is not unique of nitrobenzodiazoles. There are other fluorophores that would have similarly large Stokes shift. You can also see that the bandwidth is broader, which is also, again, another feature of these nitrobenzodiazoles. And these are things that are really difficult to change given a chemical structure. You can tune the color, you can tune the brightness, but often the bandwidth and the Stokes shifts are a bit harder to modify because they are inherent to the chemical structure.
Yeah, for sure. Another rather physical question: are these POT-M labels also applicable for classical thread experiments?
They could be. I think that you would need to find, obviously, fluorophores that would match the excitation emission of either the sulfur or the selenium one. And I think that there's no reason why they could not be if you find a pair. In between them, like the sulfur and the selenium as a thread pair, I don't think that they would necessarily work very well because none of the two emission spectra overlap well with the absorption spectra of the other. But there's no reason why you couldn't use one of them and, let's say, another fluorophore as the complementary thread pair.
Mm-hmm. Okay, and the rest of the questions go rather into the peptide synthesis.
Okay, I'll ask Fabio to get a bit closer then.
Yes, so in fact, the one came in a bit before Fabio already went a bit more into detail to that. So are those amino acids compatible with the standard protecting groups and standard resin conditions? And are they also compatible with microwave conditions?
Yeah, they are. So we have shown the chromatographic stability for the selenium containing POTEM. The sulfur containing POTEM, we also test stability. It's slightly less stable than the selenium, but again, over 85% stability, I would say, in piperidine 20%. 95% EFA, and also condition for removal of side chain protecting group. In microwave, we have run tests, and both amino acids are quite stable. Microwave radiation, 1 minute, 90 degree, 20% piperidine, and we've also tested the coupling condition, 90 degrees, 2 to 4 minutes, in the presence of excess of oxima and DIC. And they are all quite stable. The selenium containing one, again, remarkably stable, over 95%.
Okay, that sounds really good. And what about the pH stability? So is there a time limit for the stability of the amino acids in media such as TFA and DCM?
Long-term stability, I don't think we have run, but in, I think, up to a few hours, they can be stable, particularly again the selenium. Up to high concentration of TFA. If we are talking again TFA DCM, like the classic acidic medium for peptide synthesis.
Okay, so, and one last question I found is that was to the CD spectra you were showing that the fluorogenic amino acids do not change the structure of the peptide, but in the CD it looked as if the helicity was even higher with the fluorophore containing peptide. Was that just a concentration effect?
Yeah, that was a concentration effect. I think that the purpose of that experiment was rather qualitative, and we wanted to make sure that the 101A peptide was just retaining that alpha helical structure, because without that alpha helical structure, it would not bind to the 101B. And that's why I put two different concentrations so that actually the audience could see that there was the concentration was not matched, but qualitatively we could see that the alpha helical secondary structure was retained after labeling, which was the main point of that experiment. Yeah, I don't think I wouldn't dare to say that the POTEM amino acid would induce any secondary structure on its own. It would be something that would need to be explored by any peptide sequence, but in any peptide sequence, specific ones.
Okay. So then, as I see no further questions, dear Fabio, dear Mark, thanks again. It was a pleasure to have you as our guest speakers.
Thank you. Thank you very much.
Yeah, to the audience, if you have any questions left, do not hesitate to contact us also afterwards. We will forward those questions to our experts. With this, we would like to close our online workshop. Please excuse us for the technical issues we had at the beginning of the expert presentation. By this, you can see this is really a live event. Now, as a follow-up, we will provide the recordings of this workshop. Thanks again for joining us. Thanks to all our speakers, and also a big thank you to our QA manager, Klaus Helm, for taking care of the technical support. We hope you could collect some helpful ideas and information from this workshop, and you can gain some interesting own experiences with this elegant labeling technology. As you have already noticed several times during the talk, what a surprise, you can also acquire the FMOC protected NBTD and NBSD labeled amino acids from Iris Biotech. So thank you for joining us today. We're looking forward to meeting you again in the next workshop in June, which will be announced on time on our homepage and via social media. Enjoy a beautiful springtime, stay healthy, and hopefully see you soon. Goodbye.
Bye.
Bye, everyone. Thank you.
Bye. Thank you.